Hi students, let's start with new topic, that is genomics. See, first of all, before starting with this topic genomics, we should know what is the meaning of genome. See, genome, this term is given by the scientist H. Winkler in the year 1920. Now, what is this genome? This term is given by Winkler. But what is genome? Genome is nothing but total constitution, total DNA constitution, whatever the things present inside the DNA, or you can say genetic information present in DNA, or you can say one complete set of chromosome in haploid organism. Now what is this? See, haploid organism means it has only one set of chromosome. We are diploid organism, means in our body we have diploid or two set of chromosome, but in haploid organism there is only one set of chromosome. So whatever the DNA constitution or whatever the genetic information is stored in one complete set of chromosome, that is called as genome. Okay, now what is genomics now? This genomic term is given by the scientist T.H. Roderick in the year 1986. Okay, now what is genomics? Genomics is nothing but it is a branch. It is a branch means it includes the study of genome. Okay, now what exactly is there in the study of genome? Here three things are included in the study. First is analysis, second is sequencing, and third is mapping. ASM. What we are studying? Genomics. Genomics is nothing but it is the study of genome which involves ASM. Analysis. Analysis means what? Here in the study, no, analysis means to examine what type of genes are present. Okay, in the DNA genes are present, no, so what type of genes are present in a particular segment of DNA or in the gene, that is studied here. Second, sequencing we have to study here. Sequencing means what? The order in which the genes are placed. What is the sequence? How the genes are placed, that we have to study. And third thing is mapping. Mapping means what is the position of genes on the chromosome. So here genomics means it is the study of genome which includes analysis, that is examine what type of genes are present. Second, sequencing means what is the order in which order genes are placed. And third one is mapping, means how the genes are positioned. Now, next one is there are two types of gene. First one is called as structural gene genomics and another one is called as functional genomics. This genomic study, there it includes or it is of two types. One is called as structural genomics and another one is called as functional genomics. See, structural genomics means again it involves three steps. ASM, same thing. Analysis means here just now we have studied what is analysis. Analysis means to examine. Sequencing means to keep those things in proper order. And last thing is mapping, means to see the position of the genes on the chromosome. So this is structural genomics. In functional genomics, here it deals with the study of only functions of genes and their expression. So genomics is of two type. This study is of two type. One is structural genomics where only analysis is done, examination is done, sequencing is done, genes are arranged in proper manner, and mapping of genes means here it is studied that how the genes are positioned on the chromosome. But functional genomics means it deals with the functions of genes and the gene expression. Now, application of genomics. Applications means what are the advantages, what is the significance, or where this genomics are used. See, genomics are used in agriculture. How it is useful in agriculture? For the development of transgenic plant. Now what is transgenic plant? Transgenic plants can be grown with more desirable characters. Okay, so that type of plants are called as transgenic plants, which includes where the plants can be grown with a desirable character. So that is called as, so we can say genomic is useful in the field of agriculture. Second, it is used in the development of genetic markers, which is useful in forensic analysis. In forensic department, no, genetic markers are used to cut the segment of DNA. So here development of genetic markers can be done with the help of this genomics. Means genomics play again important role in the development of genetic markers, which are useful in forensic analysis. Third one is it is useful in the introduction of genes in the microbes to produce enzymes, therapeutic proteins and biofuels. I'll give you one example for this. If we have to produce insulin artificially, what we can do, we can introduce particular segment of DNA, that is certain genes we can introduce in the microbe, or you can say in the bacteria, and later on that genes inside that microorganism can give rise to insulin. So here it is useful in the introduction of genes into the microorganisms, so that particular microorganism produces enzymes or proteins, etc. Okay, so these three are the application of genomics. Now next important topic is Human Genome Project, HGP, Human Genome Project. Now see, this Human Genome Project is started in the year 1990, and it was completed in the year 2003. Means this is the very huge project. It was mega project under the International Administration of Human Genome Organization, HUGO. HU means human, G for genome and O for organization, and it was coordinated by US Department of Energy and National Institute of Health. This mega project was again help, or it is coordinated, or the partners were UK, France, Germany, India and China. So Human Genome Project is a vast project, mega project. It took near about 13 years. It was started in 1990 and completed in 2013. Now what was the aim of Human Genome Project? These 5 aim we have to study. How you will keep this aim in mind? This is the keyword MSDTT. MSDTT. Now what is M? First, what is the aim? What is the aim of Human Genome Project? That is HGP. First aim is mapping of entire genome at the level of nucleotide. Mapping of entire genome at the level of nucleotide sequence means here the analysis of entire gene is done. That is, what is the sequence of nucleotide? What type of nucleotides are present in the genome or in the entire gene? That is studied over here. In Human Genome Project, entire genome study is done. Then second, what is the aim? Second, to store the information collected from the project. What is the second aim? To store the information, whatever the project they are doing here through this HGP, they can store all the information. Third aim is to develop tools and techniques. With the help of Human Genome Project, they want to develop many tools, many techniques for the analysis of data. Then fourth aim is transfer of related technologies to the private sector. Okay, means here through this project they want, or they provide new techniques for the project to the different sectors. And last aim was taking care of legal, ethical and social issues which may arise from the project. That means there should be no dispute. Okay, and if if any type of dispute arises, it should be solved immediately. This was the aim of Human Genome Project. M for mapping, to map or to examine entire genome, to store the information, to develop tools and techniques, and to transfer related technologies to private sector, and to take care of the legal, ethical and social issues. So these are the 5 aims. You can keep this keyword in your mind. Next one, advantages of HGP. What are the advantages of such a big or mega project? First is advantage is, because of HGP, Human Genome Project, new area in biology is developed called as bioinformatics. Means what? They started, see, Human Genome Project is very vast project. All the information, it is not possible to store all the information on the paper or in a book. So here they use computer technology to store the information. Means what is the advantage in the field of biology or in the area of biology? Computer or bioinformatics had introduced. Second advantage is, due to HGP, researchers learn more about the function of gene. They got many more ideas about the genes and proteins, which has major effect in the field of medicine, biotechnology and etc. Okay, so here this is the second advantage. First advantage is, because of HGP, bioinformatics branch was introduced in the field of biology. Second advantage is, because of HGP, researchers got many more ideas about the function of genes and proteins. Third one is, because of HGP, or HGP provides complete and accurate sequence of 3 million DNA base pair. From this HGP only, researchers or scientists came to know that there are about 3 million DNA base pair, and they came to know what is the accurate sequence of DNA base pair. And last advantage is, HGP is useful for the researchers or for the scientists to study the structure and function of genes. Because of this HGP only, they got all the structures, they got function of genes, they got structure of genes. So here we have completed advantages of Human Genome Project.